thanks a lot for the invitation, and thank you for setting up this very nice event in which we bring together a little bit technology and the application of the technology, which is something that is always, for neuroscientists especially, something really fundamental. And I'm very excited today to be able to show you a little bit of our recent work on memory, and in particular on, as you mentioned before, memory transformation with time. But before we go to the hardcore science part of the talk, I just wanted to throw you off your balance a little bit. And I know that this is a scientific and a technological event, but I would like to start with something that is very close to my heart and my interest, that is something more associated with philosophy. And in particular is something that this gentleman, John Locke, said quite some centuries ago that has really inspired my research throughout my whole career. That is that memories are the essence of who we are because they allow us to be today the same person that we were yesterday. He considered memory as really a red thread that connects the different phases of our life. And in my opinion, this sort of statement that was quite bold in the 17th century really goes at the core of a fundamental question of biology. That is, how does the brain create memories that persist through time? So in the last 15 to 20 years, a lot of technological and specifically genetic advancement have allowed us to understand quite a lot about how memories are encoded or are present in the brain. And in particular, they reveal that hippocampal neurons that are active during an experience become the physical substrate of the memory that is associated to it. You might be familiar with this concept, but if you're not, I just want to touch briefly upon it because it's really important for the talk. While we live through something like today, for example, some of the neurons in our brains are active, and specifically those that are present in an area of the brain that is called the hippocampus are sort of allocated to go through a series of processes offline that we call consolidation in order to create a subnetwork that we call engram, whose reactivation in later times brings back the ability to recall the memory. So this is sort of a cycle of activity, consolidation, and reactivation, which is really fundamental for memory. And in fact, we know that a reinstatement of activity patterns that are similar to the one that were present during memory acquisition are really the essence of memory persistence. Now, this is really an important framework that has been put forward to understand memory in the past 20 years, but even longer, and that also been used to explain how you can remember things at hours, days, of months of delay from when those things actually happened. But there is something that is really sort of not captured by this type of models. It is the fact that memories are dynamic entities. With times, they change. And when I'm talking about this, I talk about two aspects fundamentally. One, the fact that when we think about the properties of memories, we know that these memories that are very fresh are very different from the ones that are encoded a long time ago. And we might all have quite some experience with this. If we think about what we ate for breakfast this morning or a few hours ago for you, we might recall it very precisely. But if we were asked what we ate the exact day 3 years ago, we might not know very well. Similar things happen also in mice. For example, mice that are trained to associate an unpleasant experience like a food shock with a specific context, they tend to forget some of the detail of this memory with time, so that they start to express this fear behavior that we call freezing, that is a generalized response to fear, in many contexts, not only in the one where they were actually trained. So we see a property of a memory that changes with time. But we also know from the work of a lot of scientists now that there is something that is called representational drift, meaning that when you look at the brain and the specific area of a mouse, for example, while this mouse goes through a similar experience with time, you see that there is always different type of neurons that are activated at different times, which brings somehow a conundrum, right, in the picture of memory that I told you before. That is, how can memories actually persist in the face of these dynamic entities? So the important questions that are put forward by this type of evidence are, in our opinion, three. How do ensemble dynamics, like the dynamic of neurons that are active, affect the persistence of a specific memory? is there a logic that drives this reorganization of neuronal ensembles that are active when memories are encoded or reactivated over time? And last, are distinct neuron subpopulations recruited at specific times to give dynamic properties to a memory? And this last question might sound a little bit specific now, but it really fits with the fact that I believe one of the most important discoveries that the community of neuroscience has done in the last 20 years is that neurons, and hippocampal neurons specifically for today, are very diverse. In fact, when we draw network like these ones, we should actually put some color on and really express the idea that different neurons have a population are characterized by different properties, be the developmental trajectory of this subpopulation, their connectivity patterns, their ability to fire a response to stimuli, their intrinsic properties, or something that we are exploring now, their function. And so we could put forward two different models for how this diversity and memory might actually interact with each other. One model being that memory might not really care about this diversity, and the neurons that are activated to support the encoding of a memory, the recall of a memory, are always the same or have the same properties, or that instead neurons really do care about, memory really does care about diversity, and the neuronal population that are recruited to support memories that are recently encoded or remotely encoded might actually be different. So this is sort of the two hypotheses with which we started this investigation. Also helped by the fact that the area we're interested in, the hippocampus, has been actually discovered to create this huge diversity of different subpopulation among excitatory neuron during development. And in particular, one thing that we and others have really put forward is that depending on the time when a neuron is born, is gonna have some different properties. So that neurons that are born at the same time or on the same day are gonna be much more similar to each other than neurons that are born at different times. So for us, really segregating neurons based on their birth date during embryonic development is really an approachable tool to identify and study difference of population. So we decided at the beginning to turn this question around, right? How do memory take advantage of this diversity, and how do they change with time? By asking a very simple question, that is, what is the functional contribution of hippocampal neurons with different birth dates to memory dynamics? This, because for us, this was a very tractable question from an experimental point of view, and actually so tractable that we could resort to a technology that has been out there for at least 50 years. It is mapping recruitment of neurogenesis-defined neurons through the injection of a very standard molecule that is a thymidine analog and basically label neurons at the time of injection. So what we did in the hippocampus is that we labeled neurons born at different time during neurogenesis, during development. And in this presentation, we will focus mostly on the early born neurons born in E12 and the late born neurons born at E15. And then we wait until the animals were adults and we put them through a paradigm that would allow them to encode the memory. And this paradigm is the contextual fear conditioning. We place an animal into a box, we let this animal explore and then deliver some food shock so that it will associate this negative experience to the context when the experience has happened and create a memory that will persist in the brain for a certain amount of time so that when the animal is put back in the context, it will release this sort of fear response that we call freezing, right? But at this time, we can go into the brain of these animals in which neurons are labeled based on their birth date and then ask ourselves if neurons born at different time during development are activated or recruited to support memory processes at different time throughout a memory's lifetime, right? So when we look at the whole hippocampal network, and here we're focusing on CA3, we see this sort of pattern of activity. We see that there are neurons, which we usually identify for the expression of an immediately gene that is called CFOS, that are recruited or activated to support memories at any time after this memory is encoded. If you wait 1 day, if we wait 1 week, if you wait 4 weeks. But the question came, are these dynamics somehow different if we segregate neuron based on the birth date? And of course, because we are here, the answer is yes. When we look at the activation or the recruitment of this neuron at baseline condition upon memory activation, we see that both populations are recruited into this memory ensemble, this subpopulation of neurons that recall and support memory processes with the same properties. But when the memory is recalled for a short window of time after acquisition, the neurons that are born late during development are the ones that drive these hippocampal processes of recall. While in a series of a sort of a swap positions, over a few days, this sort of balance will shift so that by the third day onwards is the early born neurons that are basically the one responsible for, or that are ones that are recruited for memory recall, right? And this is, of course, something that we test is very specific to the encoding and the progression of a specific memory. It does not happen if that memory is not encoded. And this for us was striking, right? There is a divergent dynamic of population of neuron that we distinguish for the birth date in order to support possible memory processes, which really raised an interesting hypothesis. That is that there might be parallel memory traces in the hippocampal network when we encode the memory. What it means is that when we encode the memory, we don't create one copy of this memory, but we create multiple of them, each in its own specific subpopulation of neurons, with a late-born neuron-like memory trace, which is reactivated for a short period of time after acquisition during recall, but not at later times, and another trace, which is the early-born neuron-like memory trace, which is reactivated if recall happens at a longer time after acquisition. Now, this is, of course, something very interesting. It was an hypothesis that could be tested for us, but to test it, we needed to look at a different type of technology to visualize our neurons. And the type of technology that we decided to use was calcium imaging. So we resorted to use miniscopes in order to basically record activity dynamic from this hippocampal network while the animal was learning an experience. And in order to be able to look and see if a neuron was activated during a specific phase of memory processing, like the acquisition or the recall, we decided to first do a baseline recording that we take as a sort of a baseline to account for this representational drift that people have seen in the hippocampus, and then look at the relative changes in firing activity of the neurons across experiences. And basically, when we did this type of analysis and we were able to record from many, many neurons at the same time, we could observe that if we look at individual neurons that are active at the moment in which a memory is acquired, like the engram neurons that I was mentioning during the introduction, which is something very interesting, that is that there is a population of neurons that indeed follows that type of dynamics that we saw with our BRDU and FOSS experiment, meaning that these neurons are active at acquisition, but they're also active at one day, and then they become inhibited when the memory is recalled at later time points. While there is another population of neurons instead that is active at the moment of a memory acquisition, it becomes somehow inhibited when the memory is recalled at one day, but then is activated again at later time. And here you just see a quantification at the single cell level of this type of dynamics and an overall morphological quantification at the level of the whole population. But this didn't still tell us if these two profiles that we observe in the divergent activation of neuronal ensembles are actually really instantiated in the early and late-born neuron that we identified through BRDU. So for that, we really needed to jump to another technology that became available to us in the past years, that is dual color miniscope imaging in freely moving animals. And for us, this was really a game changer because it allowed us to be able to record at the same time neurons that are born early or late during development that we tag with an in utero and in viral approach that I'm going to explain to you in a couple of slides, in order to distinguish the activity dynamic of these two subpopulations in the whole CA3 network, so that at the same time we can record large population of early-born neurons, late-born neurons, and also compare the activity dynamics of these neurons to the whole of the overall network. And this gave us very important insights into the biology of these two populations, because first of all, it told us that even at baseline condition, these neurons are very different from each other. Namely, while the early-born neurons are very chatty and they exhibit activity rates that are higher than the average population of CA3 neurons, the late-born neurons are instead quite silent. But still, when they talk they like to talk to each other. So late-born neurons like to be more correlated to other late-born neurons than to the rest of the population. And the same thing for early-born neurons, really highlighting how a network that we have considered as homogeneous for many, many years might actually be parceled in parallel circuits for information processing. But the important thing that it really told us, this type of recordings, and I'm going to cut a very long story short here for sake of time, is that indeed those two profiles of activation that we saw in the general population are clearly driven by different dynamics of these two populations, with the early-born neurons that are active at acquisition, but that silent at one day and reactivated again at remote recall, and the late-born neurons instead that are activated at acquisition, also activated at recall at one day, but then inhibited at recall at a later time, which is something that was really, really important for us to pinpoint to specific population at specific time. And this, again, is a quantification. The last message that is going to be really important for the continuation of the talk is that these neurons seemed also to be very much involved in a different type of response to learning, which is a plastic response to learning in one case, which is driven by the late-born neurons, meaning changes in connectivity structure in the network functional connectivity, which are short-lived and reflect the usage of these traits, and instead more long-lasting changes within, and more rigid but long-lasting changes within the early-born subpopulation. So with this, I will conclude this part, and I guess that for the sake of time, I'm going to go quite fast through the second one, just suggesting you that through the ability to really visualize activity from distinct subpopulation of neurons that we label based on a developmental criterion through an in utero injection that is directed to the lateral ventricle of the embryo, we are able to understand that there are complementary recruitment dynamics for earlier late-born neurons during a memory's lifetime, and that these dynamics are specific for the encoding and recall of a particular associative memory and end up being the seat of different memory traces. One, a plastic trace, which is instantiated in neurons that are born a later time during development, and another one, a rigid trace, which is long-lasting and instead is instantiated in the early-born subnetwork. So this is the working model that we have tested with follow-up experiment that, if you're interested, you can read about in the publication, in which we hypothesize and actually prove that there are multiple traces, memory traces in the hippocampal engram, as if any memory that we have might have at a certain point of their life multiple versions from which the hippocampus can choose from. And in particular, two of these versions are really instantiated into population of neurons that are born at different time during development, with a very transient and short-lived late-born neuron driven memory trace, which is actually responsible, and this is something that I'm gonna shorten because of time constraint here, is actually responsible for the possibility of a memory to be sort of dynamically updated if new information become available. Our memories are not set in stone for a certain period of time, and until the late-born neuron trace is actually active, we can combine information across multiple learning episodes in order to strengthen or reinforce memories that we have created. Once this window of activation of the late-born trace is actually gone, then a new memory trace takes over. This is the permanent, we call it permanent, but we know that it's long-lasting early-born trace, which is a little bit slower to rise, to be encoded, and definitely more rigid, and actually actively prevents this memory from being updated, really allowing your memory to sort of resist through the passage of time and through a dynamic environment. And of course, the consequence of this is that if we silence the late-born neurons, we are able to actually prevent memories from being committed to this long-term storage. But also, if we try to instead increase the activation of this late-born neuron, we might work to extend this plastic window for memory integration a little bit longer than what is actually usually allowed. And for that, I'll just go back to the last summary to basically summarize a little bit the short survey in the study that we published last year by taking advantage of dual color miniscope imaging in freely moving animals that allowed us really to distinguish activity dynamics from different population of neurons at the same time, we were able to show that memories are indeed encoded into multiple traces within the hippocampal network, and then these traces become the substrate for processes like memory updated or integration, which are very short-lived and rely on the activation of specific neurons that are coincidentally or not, this is something that we are now investigating actively, born at late time during embryonic development. In particular, I'm very excited about an implication of this type of discovery, that is that a memory might come with a sort of a transient trace that might work, this sort of late-born neuron trace that might work as sort of a memory buffer to be able to integrate information that we might acquire with different instances. With that, I really would like to thank the people in the lab that did this work that kept us, wondering and excited for many, many months. And in particular, 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗧, my first PhD student, who now is back in Norway and who has been really spearheading these experiments from the beginning. And of course, you for your attention. And I hope that we have time for some questions.
Fantastic. Thank you. This is such an interesting discovery and contribution to our understanding of memory. We have at least 10, maybe even 15 minutes for questions. So please feel free to ask anything you're curious about in the chat. The first question is definitely enthusiastic about neuroscience technology and maybe even thematic with this Bruker Summit. Thinking about these early-born and late-born neurons, do they differ in their transcriptome and their genetic profiles? Has anyone explored that? It's a big question.
Right. It's a huge question, and I hope that in a few months we're going to have the answer, because there is one thing that I couldn't go through very much thoroughly, but the ability to actually access this neuron based on the birth date came from something that another type of technology that we also developed. There is sort of some sort of targeted injection that we do during the embryonic development in the pup's developing head that allow us by viruses to basically label distinctively these neurons in a different way. And this is unfortunately for now one of the very few ways by which we can separate neuron based on birth date. So to say that the ability to sort of purify a sample of early-born neuron or late-born neuron became available to us just very, very recently. But there is encouraging data from the fish, from the frog, actually, and now also there was a preprint a couple of days ago from monkeys that says that there might be a very large segregation in terms of properties, but also in terms of transcriptomes for this population. We have the experiment of going to do single-cell sequencing and try to understand the trajectory of development of this neuron. And of course, the idea would be that if we have molecular tools to actually access one or the other population, I had to go very fast through the last experiment. But we might have a tool to actually stimulate the activity of this late-born neuron and maybe increase plasticity in case of aberrant memories, like those memories that are very intrusive from the beginning in case of PTSD, or memory, or maybe even reducing the plasticity of this memory in case in which instead, we have some sort of hyperplasticity pathology. So we're very much interested in understanding the genetic differences among them. Yes.
That's great. So this labeling technique was really instrumental in getting the handle on these two populations of neurons. And I love the way you've described where this could go and actually towards manipulating them. We heard a little bit about manipulating specific ensembles in Kevin Mann's talk just earlier in this session. So that's very cool.
Yeah.
So in the CA3 network, you mentioned that it was important to be able to image many neurons at the same time. Right. I wonder if you can comment, you started with the single color miniature microscope imaging. What was the typical cell yield population there? And then another thing that I've been wondering about is how many of these early and late-born neurons are there? Are these,
right,
did it become dominant in the population, or are they really still a small subset? So just to give a sense of scale and who's making up the chorus here.
Right. So that's also a very interesting question. I'll just go for the experimental details, right, of the yield that you were mentioning. Um, you have to imagine that like there is, of course, the yield of neurons that we can image simultaneously in the whole network goes between 300 and 900. Like this is usually the ballpark of what we have. But along these neurons, actually, the hippocampus is very smart, and it doesn't use that many neurons to actually create sort of a map or a memory or a representation for a single event, because otherwise we would fill other capacity very fast, right? And instead we call it orthogonalize, that is to use different set of neurons for different experience in order to create memories that can maintain distinction. So usually only 1 or 2% of these neurons are actually the ones that are really, really activated specifically to that event. Now, if you take that, the neurons that we call early born or late born, the total population of neurons is around 20% of all the CA3 neurons. They also become sort of a 20% in baseline level of the one that we can record active at a specific time, right? So in the end, what we end up in our best experiment is to record from 50 of these neurons simultaneously, which is already quite a large sample for us. And it's only possible because we really do calcium imaging. We know that there are other groups in New York and in Marseille that are doing more recordings, like electrophysiological recordings with optotagging. And unfortunately, there we always share our pain and experiences with this. The yield is very, very low. So in the end, there is a very small fraction of neurons that are responsible for this type of processes. And it's, in my opinion, like a very clever design to allow us to encode many, many memories as we live through life.
Yeah, thank you for the comments on the differences in approaching these tiny cell populations with imaging versus with electrophysiology and methods like optotagging. I've heard similar comments. It's really tough to get the cell yield to be able to draw those conclusions.
Right.
We have a question on a slightly different theme here. Do you think that maternal stress could disrupt the balance of these early born, late born neuron dynamics in mice that are born to stressed mothers?
Right. That's a fantastic question. So I have debated with myself if I should include a little bit of the developmental side in the talk of today, and then I realized it would have been a bit tight. But I'll tell you just a little bit of a teaser. Of course, one thing that is very different between the early or late born neurons is the time during development when these neurons go through the things that neurons go through when they develop, you know, like they make contact with each other, they start to create synapses, they start to connect with other areas of the brain, and so on and so forth. So of course, for us, one interesting thing was, let's go at the early stages of life, and let's see if there is a difference there in the ability of this neuron to be recruited, right? Because it might also have consequences in the way in which not only that early born memories are formed, and we find differences. Everything goes through the early born neurons at the time, not the late born neurons. But also, as we were mentioning, like we know that early trauma is actually quite enduring in the ability of an individual, even a mouse or a human, to actually function cognitively for the rest of its life. So we're really interested in understanding if like there is some sort of interaction between the time during development when things happen and the maturation state of the hippocampal network on itself, particularly looking through the lens of the differential maturation of this subpopulation. We don't have data yet, but there are a lot of arrows that point towards interesting observation.
This is also easy to sort of anthropomorphize and think about in our own lives too. This is another question about the two populations, the early and late born neurons. They ask, are different layers of the CA3 inclined to have early versus late born neurons? Do you expect there to be differences in upstream and downstream targets of these neurons? You mentioned that they're networked with each other, but what about up and downstream targets, anatomical organization? Can you comment there?
Yeah, so we sort of went to CA3 for more memory-related, let's say, reasons. But we sort of made a little bit our life easier also because there is not layering. So so if we target like a field of view with our miniscope, we are able to pick up both population intermingled with each other. However, where there is a layering is in CA1, just one synapse downstream. And in that case, the deep neurons, what people call deep CA1, are the early born or enriched in early born neurons, while the superficial neurons are enriched in late born neurons. So there seems to be some sort of stratification similar to what we find in cortex of anatomical location versus the birth date. And about the connectivity, it's actually interesting to see that the 𝗧𝗧𝗦𝗧𝗥 group in Marseille has already mapped connectivity from CA1 to the other areas of the brain, and they find interesting differences with the prefrontal cortex, with the nucleus accumbens and the amygdala that are also stratifying based on birth date. But there is very little known about the input to this population. And because we are looking at the recruitment, so the activity of this neuron during memory, we really believe that that's where the major differences in this involvement in memory through lifetime might actually come from. So we are using now rabies strategy, always leveraging this sort of genetic tagging based on birth date, right? To combine with viral methods to actually trace the connection toward these neurons. And I hope that we're going to have an answer soon about that as well.
Yeah.
In these studies, you did a lot of multi-session recording over days, imaging over days. I wonder if you can share with the audience what tips, tricks do you have for really maximizing your ability to interpret and identify, are these the same neurons? Is this the same population over time?
Right. So I'll share something that came actually from us testing the MView system from Microscopy, actually, right? So, if you want to, one of the first things that we found ourselves struggling with was that when you use a lens like a green lens, you have chromatic aberration in the green light and the red, in the green signal and the red signal, right? So you have to really understand how these two align with each other. And so a postdoc in the lab, Maria, that was working with 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗍, she came out with an idea that, in my opinion, was really, really smart. It is, like we usually try to align our field of views on the two photon, et cetera, through the blood vessels. Why don't we inject a red dye into the blood through the tail vein in the mouse to just try to understand in the two channels what is the optical relationship in a Z stack that we record between the two? So how further apart the green optical plane of imaging is from the red optical plane of imaging, right? And so we did this very systematically. It's very reproducible. It was around 70 microns for us. So we basically went through this, and that guided the experiment. At that point, we also realized that since we have the same signal in the green and in the red, and one is very stable, that is really the blood vessels, we could use this signal to actually align field of views across days. And so we basically also use this trick to sort of inject the red dye after the imaging has happened sometimes to also get this sort of alignment. And then use the red cells in order to align, which are static, right? And are very stable across time, in order to align across days. And so that's how we could actually get a yield of alignment that goes between 30 and 40% of the grid course.
Yeah, thanks for unpacking that. It's clear that the second channel was a big assist in being able to do this alignment and tracking. All right, we have one more question here. And again, if you're in the audience and wondering, we can probably fit in one more. So feel free to enter it in the question box. They ask, if the number of neurons related to a single event is so small, something we talked about earlier, how can we target those neurons effectively? And are there clinical applications that would be better suited to this targeting? What do you think about that?
Yeah, so of course, in mice, we have quite a few tools to target the neurons that are active specifically when a specific memory is done, right? We can use some sort of structural criterion, if you want, like the birth date or the expression of the genes, but it sort of gives us a population of neurons, not really the one that are active at a specific time. But actually, there is a lab in Stanford, I think it's Alice Ting, that developed a series of tools that can allow, in the Dysart lab, of course, that allowed us to exploit the fact that neurons that are very active at a high concentration of calcium, with the possibility of inducing some changes in conformation of protein through light, for example, to basically like combine these two criteria, and then only upon delivery of light and calcium increase, you have some sort of genetic recombination that labels those neurons specifically. And so you can manipulate, kill them, see them whenever you want at later times. So of course, this is very invasive, and it's very difficult to you know, do this type of approaches in humans, because I guess that's where the question was going. But in my opinion, the more we understand about the type of molecular changes this specific population of neurons responsible for a specific memory goes through different times, you know, during the life of a memory itself, from when the memory is born to when it matures to when it becomes old, the more we might find target to actually you know, very non-invasive targets to actually manipulate that. So for example, we have another study in the lab in which we basically like try to bring back a memory, a traumatic memory that seemed to be forgotten. And by using this type of activity dynamic tools, we have revealed that there is a window in which if we teach the animal something very good, the type of reinstatement of this trauma that would basically carry over and become a memory, a full-blown memory, even though the brain had actually suppressed that, can be prevented. And this is just very little invasive, and it comes from understanding when these neurons are active and what type of genes they express. So in my opinion, if we can align those two streams of information, activity and genetic expression of specific markers, we might come up with tools that even in a non-invasive way allow us to manipulate these few neurons.
That's just a stunning concept. If there's any Hollywood movie producers watching today, maybe that could be the next full-length film. Thank you so much, Flavio, for sharing your research today.